Hello.
Hello.
Hi, how are you?
I'm doing fine. I did a physics binge yesterday on YouTube. So I'll put the title of one of them in the chat because it was about neural nets. And I was wondering if you could program a tensegrity as a neural net or a neural net as a tensegrity. So I'll just put it in the chat.
Okay, it sounds good. Yeah, I have some interesting things about that later, but yeah, we'll have to look at that. Thank you for the interesting update there. Okay. So hello, Barshan and Varshish and Shreyas. And Jess and Morgan.
Hey, hi.
Hi, how are you?
I'm fine. How are you?
Good, good. All right. So does anyone want to present or talk about anything today?
Oh, I have a nice paper too, if you want to see it.
Yeah. If no one else wants to present, why don't you pull that up?
I'm still working on the hyperbolic spaces thing. So converting the GN into the hyperbolic space. So once I get substantial results, which I hope in like couple of days, I'll just present. I'm still working on it. So yeah.
It's fine, yeah.
Hello, am I audible?
Yeah.
Okay. So I did some research on the set attention and incorporating attention into the lineages, and I figured some things out. Actually, I scribbled on my notebook, so I didn't really prepare a doc. I like to write everything down while I research. So I'll just give a verbal update.
All right.
So there's this so the paper incorporates two types of algorithms. Like one is node set attention, which is mostly concentrated on the nodes itself, like the cells, which can be later used for the classification of cell fates. But there's one more algorithm, which is like et set attention, which can be later used for if we can incorporate some physical or chemical properties in the cell. So as Susan said, that if we can incorporate some physical properties like tensegrity or some chemical properties, this can be a very good opportunity for that. Like very good algorithm. And it has proven results in that paper too, in the medical, basically medicines data set.
Okay, that sounds good.
Yeah.
Yeah, I think we talked about, if you're familiar with Layla's work from last year, and you can see some of those recordings. We talked about this sort of attention last year, but I don't know what the state of that is. If he, how much progress he made on, because it was like we were going over a lot of the reviews of a couple of papers where they kind of lay out some of this stuff. And then they didn't, it was just kind of like a part of that paper. So I'm sure there's a lot of stuff that people have been trying to do with that, but then, getting it to work for what we want it to work for is the thing. But yeah,
exactly.
Yeah. So yeah, I look forward to that.
I'm trying to prepare a whole pipeline, with the data sets we have available and what I can build with it. So finally, I'm looking forward a tool which I can build, like the worm web, where we can take the visual 2D image or the 2D diagrams, and then we can select each cell in that, and we can understand that how it affects the cell fate of the final cells that we have in the lineage. And that can also be incorporated as a 3D design for the visualizations. So that's what I'm thinking about.
Okay. Well, that sounds interesting.
As a final tool.
Yeah,
exactly.
Yeah. Yeah.
Thank you for
great.
listening.
You're welcome. Yeah. Okay. Well, thank you, Barshan. And it looks like Susan put in this link. The brain's learning algorithm isn't backpropagation. So this is a YouTube video. Okay, so that's, because backpropagation is something that they made up in neural networks, and it was a way to sort of, it's not really feedback, but it was sort of a way to introduce a feedback mechanism. So, but it's not actually feedback in the conventional sense. It's just a way to update things and then have that. But of course, the brain doesn't necessarily do things like that. The brain has a recursive structure, but it has a different, unknown to us algorithm. I don't know if it's necessarily an algorithm. It could just be like random updating, and then it sort of is, emergent or something like that. I don't know. Looks like Utkarsh is here. I think you posted in the Slack. So I was interested in hearing more about what you were talking about in the Slack.
Hi, I'm Utkarsh from IIIT Sonipat, India. I'm an undergrad student from second year, and I recently attended a hackathon at IIT, the chance to work on biologically inspired language models from Pathways. You might have read it. The Dragon Hatchling is the paper. I became really interested in working more about biologically inspired AI systems, and I was researching this weekend and actually implemented one diffusive spatial graph, which I wanted to show.
Okay. Yeah.
I just wanted to flex my drawing board today. I didn't prepare a presentation. Instead, I just pasted my brainstorming board. So I was just trying to understand how the biology works for our case. I started from the lineage or the embryogenesis, how a single cell breaks into back and front, then what the paracrine signaling is, if that's the right pronunciation. I understood that the cells communicate by sending a chemical signal around them that diffuses over time, with distance. And then there are some physical mechanical constraints like the pushing and pulling nature, which increases, like the tension and the contraction increases as it ages. So right now, what we are doing is, I believe that we are using graphical neural networks, right? And I found like I was questioning why we are using this, and I believe there are some errors we are facing here. We are assuming that the connected neighbors to a node or a cell has equal importance irrespective of their distances or their physical contact. And we are just using agency matrix that is calculated from these connected neighbors to answer our questions, right? So the problem we are facing with the GCNs are that they are black boxes. We are not able to answer our biological questions. Like we can predict whether a synapse will form or not, but we cannot answer why would it form, right? So I was just saying that we shift to DSGs. In DSG, I say we start from one node and treat it as an agent. We put some information, the initial information, like the coordinate in a 3D space, its gene expression, and the volume or the actual space it's taking, right? Then we calculate a chemical tensor that stores the strength of chemical interaction, which will follow a distance decay, which I was talking about. This will be calculated using a morphogen gradient, which I'm calling tau here. This calculates how the morphogen field forms around each cell, and this tau actually is the decay constant. Then I'm also using an attention parameter, which will actually check whether whether the ligand secreted from a cell can actually be smelled, or we can say the chemical can be interacted with by the second cell. We are talking about the different one, whom we are trying to send the message. Apart from this, I also created a mechanical tensor, we'll say cellular tensegrity, which will answer whether two cells are like physically stable enough to form a connection. I'm using Hooke's law for now, which is like simpler. And now the important question is how the learning happens. So in neural networks, we use weights, right? And these weights are formed by backpropagation. And after like 1,000 and 2,000 iterations, these weights are like impossible to understand. They are just random features that we don't understand typically. So I say instead of adjusting the weights, we try to adjust these two constants. In our case, the attention, not the attention, like the morphogen gradient tau. And the second one is for the mechanical tensor, we are using the K in our Hooke's law, right? Which calculates how the cell's membrane will interact. We will adjust these. So one way to adjust them was we can manually change them, but that would just make it that would just remove the AI part out of it. And the other way was to use the normal neural networks way by iterating over the error and cost functions. So instead, I just read about Kajal's principle of metabolic cost. How we do this, we assign metabolic energy to a cell, and how much energy a cell uses is there's a maximum cap, or we can say there is a threshold to how much energy a cell can waste upon. And using this, we can calculate the reward function for that. This will help us calculate the edges between two cells. And for example, if we say that a cell is at, the two cells are around 10 centimeter apart, so that would use really high metabolic energy, and we'll have to penalize the connection, right? This is how I'm trying to fix the tau and the K. I did actually have some results which I will share later. Once again, I tried to answer some questions myself. So I was just questioning, is neural network or the back propagation, it is all just curve fitting, right? In multi-dimensional system, in thousands of dimensions, we are trying to fit a curve. Is it truly intelligence, or how humans function? So there are a few problems with my approach in BCG. What if our biology is incomplete? What if there are things that we don't know about, right? There can be new researches, there can be new discoveries tomorrow. What if instead of there is another thing that the cells communicate with? Maybe bioelectric signals I have mentioned here, right? Mm-hmm. So in these cases, TSGs break because we are basically telling it what are the possibilities, and we are trying to understand how they change when a synapse forms. We can answer questions, but we cannot answer things that we don't understand yet. And in such cases, GNNs are really great because they work through the data. They can form these features. I would say a bioelectric signal is a feature. They can identify these features, although we won't know which features it is talking about. But yeah, they are good at predictions. They can instantly tell what the next synapse will be, or if the next connection would form or not, right? I was researching on whether we can combine these two to build one common system, and I found it is already an ongoing research on mechanistic deep learning using GNN as a residual layer. We combine the results from both of these and set a threshold, how much they are important. So suppose our DSG says that cell 1, 2 should form a connection because XYZ, and GNN tells that it is impossible based on the data we have trained with, right? So we'll just say maybe the probability would be less, around 50%, let's say. And if GNN says that a connection should form, but DSG says that there are laws that are getting broken, then again the probability will be less. There are other missing links in DSG in my current approach or my current implementation, which I plan to fix in future. One of which is I have synthetically generated the data set. I didn't have access to. I'll be getting it today, or I'll just post it in Slack today. For now, I was generating the data set based on Susan. I was using the XYZ. Actually, this is basically, I didn't have the actual data set, and I wasn't accounting for the overall shape of worm. I just watched the previous videos and I saw someone talking about TDAs, and right now I didn't implement it, and I'm not taking consideration of sinusoidal elongation or the microscopic morphogenesis. I do have an approach I can figure out later, using instead of K, which is scalar right now, I will shift to anisotropic tensor later. And for TDA, I will add a global topological loss term. Right now, I wasn't using that. There is one more problem. Right now, I wasn't accounting for the time or the minimum threshold. Let's say a cell can only bifurcate or can only split after it ages for 40 to 50 minutes, let's say. I wasn't accounting for that. I was just working with losses for now. My current dataset had no notification of time, right? This is just explanation of how DSG's internal layers work one by one. Starting with the chemical tensor, which I talked about, I am taking the decay rate as tau here. And this is the standard decay. This outputs the values in between 0 to 1, which is helpful. Otherwise, it would be hard to monitor and update the values without it. Other than that, I'll just skip to the results I was talking about.
All right.
So the idea of DSG is to make everything like a white box, to have a proper understanding of what is happening in the data, to understand it. So this is how the dataset started with a singular cell, and these lines, these edges represent how the connections form based on the diffusion law and the distance and physical constraints. This is hard to understand right now, but we can see how the parameters evaluate, which I was talking about, the tau and the k, which are the two primary parameters which I'm trying to adjust, and these are the constants which I believe are the one that exists in biology, in actual biology. I'm trying to find the one that gives the best units for a life form, I would say. For now, for such a small data, these are linearly varying, but it has a specific reason how they are changing with this. I can I'll just run this. These heat maps, they show, they compare the physical distance versus the probability that an edge or the connection would emerge. These are really helpful if we want to compare how the distance is playing a role in forming an edge. And I'm finally combining this with the mechanical tensor to decide whether an edge would form or not. If we see the output, right. So we can see how the tau and the k varies with different epochs. We can check at what stage how it changed and why here. And at the end, we can specifically target a cell that we want to research about, and we can know why it formed a synapse if it did, why it formed a connection if it did. That's it from my side. Okay. If anyone has a question, I'm open to.
Yeah, I had a few questions. So first of all, you're proposing that, instead of having kind of weights that you tune basically two parameters, and it's tau and k. So could you talk a little bit about those, or show what those are?
The tau and k.
Yeah.
Okay. Wait. Let's talk about tau like a diffusion rate. I'm saying that if I spray a perfume right here, if I use a perfume, I'm calculating how the smell would decay. How the smell, I'm calculating the decay using the tau here.
Yeah.
Is the constant. And for k, that is just basically the spring constant from Hooke's law. I'm trying to understand the cell membrane as a spring functionality. And these two constants I'm trying to figure out with my model.
All right. Yeah, that sounds good. And so that's the behavior of the membrane. All right. Susan, did you have anything you wanted to add?
Yes. For the epithelial cells, I found that the membrane didn't play a part exactly. It was the connections that were determining its elasticity. It's where it attached at the tight junction. There was an adherens junction, a tight junction, and then further down into the cell there's desmosomes. And then it was attached to the extracellular membrane. And the attachments seemed to determine what was going on with in the elasticity, because a cell is not attached all the way along its membranes. There's hyaluronic acid in there. I don't know. They don't attach. They just have very specific attachment points along their edge, and then they're attached at the bottom if they're an epithelial cell. Yeah. So I guess you could look at the paper I just put up there. It kind of gives you an overview of a bunch of things.
Right, yeah. I believe I do have some study to do about biology of cell membranes, like the mechanical view. I have to look into it further.
Yeah, well, it's really interesting. Like
Yeah.
when an epithelial cell invades, when they invade tissue for cancer, or when they're hatching, when they're an embryo, they lose their adherens junctions so that they can flow. It's interesting. I find it fascinating anyway. So
Yes,
I hope you do too.
Definitely check it out. Yeah. I was finding it quite interesting. This is my first time learning biology since school, like 5 to 6 years after, and I'm finding it interesting at this point.
That's great. That's very good.
Yeah, that's great. Let's see. So, where's some of my other questions here? Yeah, so I like having like different parameters you can tune for, kind of optimizing the model. Yeah, definitely. There's this aspect of shape that's necessary. So we've done some work on the topological data analysis. That's what the TDA stuff is. That's we've been trying to do that for several years, and it's hard to kind of know exactly how we want to do that. But that's sort of the end part where you want to visualize things. You want to be able to generate some graphs and some heat maps, but also like actually map it to sort of the curves of biology, as it were. And you have like some sort of visualization where you can put things into the biology. So I mean, that's useful. That's why we kind of hit that aspect of the project, and we haven't really done a lot with it. A little bit, but it's been hard. I think Shreyas who presented on the punk array ball, and where you have these spaces.
Yeah, last time I presented.
Yeah, and so that was another a good sort of way to model the biology. So let's see. Yeah, I think, and then this. So I'm not familiar with DSGs, diffusing spatial graphs. Is this a newer technique, or is this like something I've not heard of?
Actually, I just sat down and discovered it myself. Like I was researching. I started on Friday, so I was just going through GNNs and I was asking questions to myself, like why we are doing this, why not this. I shifted from GNNs to GCNs, then I shifted to GANs, and inside attention, I was questioning why we are using attention without any knowledge about the spatial distances. And if we change those spatial distances, if we change the softmax function to something like our own custom function, that is basically what DSGs are. This is how I discovered DSGs.
Right.
I didn't read about DSGs before this. It is basically what I wanted to build, and I understood like it already exists.
Yeah. So I guess this is a question kind of in general. This is of my curiosity, but so diffusion models are kind of basically a way to deal with black box data. Is that correct? Would you say that? As opposed to attention.
Yes, I would say that.
Okay, yeah. Because I mean, yeah, we've been talking about actually, in our Saturday meetings, we brought this up a while back now, and we never revisited it. But this idea that everyone uses attention, and there was a paper, "Attention is All You Need," from about now almost 10 years ago now, and it was everyone talks about it, but then there's like, is that really the best way we can do things? And I mean, it's like limited by constraints and limited by what people know, and so yeah, I guess diffusion models are a way to really model that black box, or at least it because attention requires you to have sort of a good command of the feature space. But it is limiting for like something where we don't really, we understand maybe like in biology we understand the parts list, right? And then, but we don't understand necessarily how things work. I mean, we do, but kind of they're all like decoupled. So like you have one process here and one process here, and one of the aspects of systems biology that everyone thinks is great is that you can bring those together. Like you can describe all these processes as part of a big map, and but that requires some method that you can say, okay, I don't necessarily know what's there, what these processes. I mean, how they, can I simulate them with like a random process, or do I need to use some other technique to simulate them, or do I have all the parts in my parts list even? And that's the thing that like is limiting to a lot of the approaches of modeling biology, is that we don't necessarily know everything about it, and we don't necessarily know how they work. So it's
exactly. I was asking the same questions, like whether our knowledge about biology is already complete or not. Maybe there are things that we don't know right now. In these cases, like the regular neural networks perform better, at least for the output we are talking about. But again, the problem like we have is that we can't answer questions with it. We can't tell why something is happening. We can't just tell like it is happening or not.
Yeah. Biology is an observational science for the most part. It's and people will summarize things as, well, we think this happened, but maybe I'm wrong. I don't think they're wrong, but they say that, and it's kind of frustrating from an engineering standpoint.
Yeah. So people will make a big deal about like how stochasticity is the key, but it's like, okay, but beyond that, how do we know? Like, okay, obviously, okay, random processes are the key, but then we also have order. So how do we get from that sort of thing to order? And so that's always a thing in so people model things stochastically and all that, but it's like still, that we still don't really, it's observational. You do an experiment, you see, you get data, and then the systems biologists take the data and use that, and that's pretty much the whole thing, like there's no, well, you can do alternative generative processes or create like mutants or some other thing where you're modifying or perturbing the system, but that's limiting. You can basically do that, or you can do like genetic engineering where you can create something in the lab. You can boot up a vesicle, but it's hard to sort of get good control of those kind of systems. So yeah, good stuff. Thank you. Thank you.
Actually, what I believe from this project from DevoWorm was that we can divide the system into two parts. The one where we are trying to answer the questions that are still unanswered, but can be answered with what we know about biology. Like we know about the mechanical things, we know about the data, like the time series data, about how a cell evolves, how it breaks, how it forms connections. So, but we are still not able to answer those. Maybe we are not looking at the data correctly, but the systems are like better at watching the data and understanding it right now. This is why I built the DSG approach. On the other hand, for like exploring new stuff, like trying to find out what even we don't know, that's still like pretty difficult, I believe. Like the combining the GNN and DSG part.
All right. Well, let's move on to Susan's paper. Or Shreyas, you want to say something?
I just had a question. So, like, if you are moving, it's fine. I had a question in the work which you presented.
Okay.
Yeah, so regarding the topological thing that you were discussing, TDA. So, yeah, I get why topology is needed, but I had been also looking at like how can we include it into the model and constraints. So, like, how do you aim to include topology into the model, and how do you make sure that you respect the topology of the brain in the model? Because that's something which I was looking towards and which I thought was a good thing to discuss.
Okay, by topology you mean like the spatial awareness, right?
Yeah, there are multiple aspects as I understand topologies. One is like individual spatial aspect, and there is relative distancing. Like one is you are relating how you are surrounded with, and one is like absoluteness. So, like, in general, like what's your view on topology of brain and how
Yeah,
you
I get it.
incorporate?
Like I was reading your approach from the hyperbolical plane. I was like intimidated from the mathematics. I I thought something simpler, like how about instead of instead of understanding how the space around me looks, I would just see it. Like for in my case, I don't have to understand that the door is 5 meters apart. I don't have to like have the data for it to know it. I can like maybe watch it, like how the bats do it. They send a signal around and they can figure out the distances. They can understand the topology by that. In same way, I am trying to use the Euclidean distance and using diffusion rate with diffusion and decay rate with that to understand like how the chemical understands the topology, like calculates how it. Yeah, that's basically what I'm trying to say.
Okay, the mechanical system or the mechanical environment is also very important, not just the chemical environment.
Yeah.
It's an interplay.
Right. Yeah, interplay
Right.
is always a thing, right?
Yeah.
Yeah, I mean, I think at a stage like this, though, it's like you get something working for one thing, and then you say, or again, it's like here's the chemistry, here's the mechanics, and then it's an interplay, so we don't exactly know how that fits together, but we can try different things, and yeah, I mean,
actomyosin does fit together with chemical and physical properties of what is going on, because the actomyosin will cause contractions which will make the tissue stiffer.
Yeah.
And single cells, I wanted to point out that single cells have different properties than connected cells, than tissue. Like they are different.
right.
You always need to keep that in mind. If it's a tissue, it won't behave the same as single cells.
Yeah.
The connections matter. At least mechanically, they do.
Okay, Barshan, you had something to say?
Okay, yeah. So let's say we divide this properties like chemical, physical, and biology. So I'm guessing that biological properties are the ones which cannot be predicted much, and it's mostly observable, and it differs from cell to cell. And physical and chemistry properties can still be predicted and worked upon. Is that the till now the the biological development that we have looked through? Like,
Well, I mean, I guess so first of all, we don't completely know all the biology. I mean, we know like like in C. elegans, we know what the cells are. We know how they tend to behave, but there's a lot of there's the biochemistry of the cells. There's their mutant phenotypes or things like that. And then we know the chemistry and physics, but we don't know kind of their interaction. So if they interact in a certain way, what does that look like? we can observe things in an experiment, but those are the interactions we can observe.
Okay. So we
And
don't
then-
really know if the physical and chemical properties are related to each other or if they change each other in any sense.
Well, we know that they're related, but like, you know, it's kind of like causality, right? Like understanding causality from
Yeah,
correlation. So like if two things we know they kind of are, you know, are sort of related, that's great. But like how exactly are they related and what like so when you have two interacting systems, strongly interacting systems, what tends to happen is you get a lot of interactions and it's hard to predict those interactions. And it's sometimes
Okay.
hard to sort of, you know, understand their pattern because you can measure them and get a like a matrix of interactions. And then, you know, you might run an experiment with the strongly interacting system and you might run it like 5,000 times. And if you look
Yeah,
at the
yeah.
interaction matrix, it's always different. So if it's
Yeah.
always different, like, okay, they follow physical and chemical laws, but it's also the case that there's a different pattern of interactions. So how do we know, other than saying like, you know, other than kind of like mining that data and proposing maybe some function or something, how do we know what the actual thing that's going on? I mean,
So
how can
basically, the data till now, it's not general in every cell case, and like it's differing for each embryo, and so we are not able to generalize any properties till now.
we can generalize things. It's just kind of like you. One great thing people say about biology is there are always exceptions. So like if I have a law or some sort of function, mathematical function, there's always going to be like exceptions to that. And part of that is because in complex systems and strongly interacting systems, you get these interaction matrices that are always going to be slightly different. So we know kind of that there's a trend, but we don't know, we can't make an absolute prediction from that. It's just always going to
Of course,
vary so much. Yeah.
Also, is there any data set available where they have recorded the physical and chemical properties throughout the development process?
Well, I mean, C. elegans, there, you know, of course, we have the cell level data, we have gene expression data, we have peptide data, we have other things, and it's, yeah, I guess there are physical maps of interactions as well. You know, and then getting the data in the same format is the problem. So a lot of people will do these kinds of experiments and they'll put out a graph and
Yeah,
try
got
to
it.
get the data for that, and it's like, well, it's very, I don't know if we can get some of those data. I don't know if we can read it and integrate it with some of the other
I mean, I still have time for a month, like before I start on working on something, so I can clean the data if I want to.
yeah
That's the thing.
yeah yeah
Like
there
I went to the GitHub repo, like most of the data were about the positional info and the timings, basically.
yeah I mean that's where we've been working from mostly.
Yeah.
But yeah, we've done some things at gene expression. It's hard to connect everything though. I mean, it's like you know, I mean, it's not something you can do in a weekend. Let's just say.
Yeah, that is true.
Yeah. So I would be cautious about that. That's why people pick their battles when they do these kinds of things. They kind of say, okay, this is we'll try to integrate these two types of data and make some inferences. But um, yeah, it's not, you know, if it was easy to build a comprehensive map, it would have been done already, probably.
Yeah, a map would be very helpful. Yes.
Yeah.
Got it. Thank you.
You're welcome. Yeah. All right, so let's move on to the. I know Susan put this paper in the chat. So this is a paper on unlocking the therapeutic potential of cellular mechanobiology.
It's a review of various things, so I just thought it would be useful.
Okay, let's take a look. All right, this is Science Advances review. This is in biophysics, unlocking the therapeutic potential of cellular mechanobiology. It's from last year. Those are the authors. And let's read through the abstract. So, mechanobiology is a rapidly advancing field at the intersection of biology, physics, and engineering. It reveals how mechanical forces shape cellular behavior, tissue architecture, and disease progression. By elucidating how cells sense and transduce mechanical cues, Mechanobiology has fundamentally advanced our understanding of processes ranging from migration and differentiation to immune responses and tissue modeling. So these advances have driven the development of innovative biophysical tools and engineered biomaterials that enable precise modulation of cellular microenvironments. So yeah, this is something we can do. We can create engineered biomaterials that can replicate things in like biology. So we know something about the material properties. We know something about this, how the cells behave. We can use things like cell culture, and we can use things like CRISPR or some sort of genetic engineering method to create unique materials or to program the materials so that they exhibit certain properties under certain conditions. And so, this is something where you can do that. You can also, use these kind of biophysical tools that allow you to measure things at finer grained scales. And then, of course, you have putting that all together, which is harder to do. So this is kind of what they're getting at here. Okay. Translating mechanobiological principles into clinical practice is giving rise to mechanomedicine, a previously unrecognized paradigm that integrates mechanical forces as key modulators of health and disease. This review highlights how mechanobiology informs therapeutic strategies across diverse domains, including cancer immunotherapy, cardiovascular and neurodegenerative disorders, and regenerative medicine. By bridging fundamental discoveries with translational applications, this review positions mechanobiology as a cornerstone of next-generation medical innovation, translating mechanistic insights into impactful clinical applications. So this is where they kind of get into mechanobiology. They talk about some properties of mechanobiology. So one is extracellular matrix stiffness. So this is a central parameter for the field. This is where you have cells, and they're embedded in this extracellular matrix of different things like collagens and other and actin, I think, and other things, proteins. We talked about these aggregates that form in this cytoplasm of proteins and mRNAs and things like that, biomolecules. And you also have cytoplasm as well. So you have all this stuff in the ECM, and of course it affects sort of the way in which the cells move, the way in which they interact, the way in which they can maintain their sort of adjacency. And this is, think about this as like a gel. So like if you were doing cell culture, this ECM would be replaced by a matrigel. And, you would have the cells sitting on the matrigel. And they would be, sometimes what they can do experimentally is modify that matrigel stiffness. And the cells will do, they'll do certain things. Sometimes they can modify the substrate as well. And so this all is to sort of look at the effects of the extracellular matrix. And the extracellular matrix has effects on sort of the mechanics of the cells, but also the biology of the cells. Because as it turns out, when a cell adheres to something, or it doesn't adhere to something, or it makes a connection, like a neuron makes a connection with another neuron, that they have different behaviors biochemically than when they don't. So you have cells, sometimes they adhere to certain surfaces and they express genes for, producing, say, like adherins. Sometimes they're on a looser substrate and they might be stressed out in that environment and they produce the stress response, which is a metabolic response in the case of cells. And that's going to make a difference in the sort of the profile of the cell, right? Sometimes, like if you have a stem cell, sometimes you can even guide its differentiation by kind of pushing the cell in different directions based on the substrate or based on the stiffness. Okay, so yeah, and then of course, this is always a thing that, maybe is associated with disease or with other types of functional phenotype in the biology of the organism. So we have all sorts of terms that we can use for this. We have stiffness, rigidity, Young's modulus. These are all things that, sometimes they'll measure just like rigidity. Sometimes they'll use something like Young's modulus, which is more sort of, it's an actual measure. So there are different ways that we can sort of understand that matrix. Okay, so that's the ECM. This is a figure where they show kind of what this looks like. So there's the substrate down here. We have integrins. We have this clutch mechanism, which allows for, the thing to adhere to the substrate. We have these things like talicins and vinculins, which accumulate at the base of the integrin and engage and disengage this clutch. So this cell can move around the substrate. It can adhere to the substrate at um, to different degrees. And then you have a bunch of things that regulate mechanotransduction, which is the transduction of forces from that substrate into the cell. So you have myosins, you have actins working in concert, and they transmit forces up to the nucleus, which then is the site of gene expression, where a lot of things are produced, a lot of genes are expressed. And then you have ribosomes, where the amino acids are created, the proteins are assembled, and then they build these things, renew them, and build other things in the cell. So that's kind of the role of mechanotransduction. It can modify gene expression, it can modify protein production, it can modify behavior of the cell. And so, we're interested in these kinds of mechanical models. So we have models for elasticity, we have models for viscosity, and for viscoelasticity. And so they show the physical parameters here. Susan?
Oh, well, and that's why and that's why I did tensegrity.
Yeah.
So that it did all three things at once. But anyway,
Yeah. So yeah, they break it down here because they want to show the different components. And then there's this interesting thing about the network. So this is the network here of different types of covalent bond. And so this just shows the difference between elasticity, viscosity, and viscoelasticity. So in elasticity, you have covalent crosslinks, which resist deformation. This is in a cross-linked hydrogel. Then viscosity, where they show chain mobility, where they show this sort of network that flows under deformation. So you have one that resists deformation, one that flows under deformation. So these are things that are, so in viscosity, it's like a lipid bilayer. In elasticity, it's a cross-linked hydrogel. And what Susan's saying is that tensegrity kind of switches between those two states or more states to maintain stability. So you can have something that's elastic, you can have something that's viscous, and sometimes those are separate things. So sometimes they're just strategies for dealing with forces and stresses to the cell. But sometimes you can have both of those modes in the same network. This depending on the material properties of it. So there are soft materials that do these kinds of things. And this is kind of one of the things about like tissues and cells, is that they exhibit these multiple states simultaneously. If you're familiar with sort of nonlinear mechanics and some of the types of materials, they're very exotic materials that people are engineering now. But things like ketchup, which is thixotropic, and it has these sort of nonlinear dynamics when it flows. And those are the kinds of things that we're interested in biology, because those are the things you kind of see in biology, things like that. And so then there's viscoelasticity, where you have weak crosslinks, covalent crosslinks, and chain entanglement. So these are all kind of where you have these multiple traits in the same network. This is a figure showing this. This is just showing the mechanics where things are activated at the membrane, and then there's this tension that's transmitted into the cell, and then you have this sort of the mechanical cues, you have ECM stiffening, and that's all transmitted into the cell where you have this mechanical sensing mechanism. So this is the cell membrane here. It's under high tension, and there's a focal adhesion. This is where it attaches to the substrate. And so this is just showing this. This is the nucleus, actually. This is the cell membrane out here. So this is the nucleus with nuclear pores. This is the DNA inside. And then these are the focal adhesions that are built with, going out to the cell membrane, and then the cell membrane adheres to the substrate. So these focal adhesions are important for keeping the cell stable or allowing it to migrate. And then you have the mechanical cues that kind of trigger that process. And so then they have these different maps: stiffness and viscoelasticity, matrix curvature, spatial confinement, and hydrodynamic forces. So they're modeling, this is, kind of like a multiphysics model of what's going on with cells, is that you have these kind of responses to physical stresses and physical forces. You have this sort of unique morphology where you have curvatures in both, in both the substrate, and then the cell conforms to that. You have spatial confinement where you have confined migration, where, you have this sort of, this is in a biological context, say where you have metastasis in cancer, and you have this sort of confined migration of the cell. And then hydrodynamic forces. So you have, in the body, you have not just that ECM, but you also have a lot of liquid. And those liquids have hydrodynamics at different scales. So you have hydrodynamics at sort of this nanoscale, which is where biomolecules are living and behaving. And then you have the cellular level, which is above the nanoscale. And above the nanoscale, you tend to have all sorts of shear forces and turbulence and other types of pressure that can affect flows in biology in the cells and in other structures. So this is atherosclerosis, where you have some sort of fat plug in the artery. You have wall shear stress, which is just stress against the wall of the artery, and then you have pressure building, behind that and in front of it. So this flow is like through a pipe. If you partially plug up the pipe, it's going to create pressure, and that's going to create turbulence downstream, and that can affect the phenotype of the heart. It can affect the function of the heart.
One really interesting thing about blood vessels is the endothelial cells in them. If they aren't under pressure and flow, they don't maintain cell type.
Yeah,
like if you're going to study these things, they clamp off the blood vessel and keep it under pressure so that they can look at it through a microscope, because it'll disintegrate if it isn't under pressure. It's interesting.
Right. Yeah. And you can think of a good reason for that, right? It's like you don't want it to differentiate away from being an artery wall. If your artery wall like disintegrates, well, that's it pretty much. But there's this sort of physics of where the cell is supposed to live, and so if the cell is there and it's exposed to those pressures, it's going to have certain roles, certain niches, certain fates that it takes on, and then it's there, and then you can actually transplant stem cells into maybe some heart muscle, or maybe create like something that you could maybe engineer arteries, where you have to kind of not just have the right cells, but you have to have them in the right environment, and so then they can be integrated fully into the organ. So this is an important thing, is this environment. It's a microenvironment, they call it, and it sort of tunes the cell to what it's supposed to do, and it maintains that sort of environment. So if you perturb it, then it can lose its it can migrate away. The cell layer can disintegrate, or you can have cancer phenotypes, whatever. So this is going through this article's review. They talk about different cellular molecular mechanisms of mechanosensing, hydrodynamic forces and shear stresses, integrins and focal adhesions being physical anchors and signaling hubs, cytoskeletal connectivity and linker of nucleoskeleton and cytoskeleton complexes, basically linking together the different structures that form around the nucleus and the cell membrane. Membrane tension is a global modulator of mechanosensitivity. Then they talk about the MAPK-ERK pathway, which is where you get MAPK-ERK is like a kind of it's a metabolic pathway. It's a stress pathway, but it also integrates a lot of those mechanical cues together. So MAP-ERK is usually associated with growth factor signaling, but it's also emerged as a key mechanosensitive module. So this is actually something that's involved in development as well. When you have different types of morphogenesis, you need growth factors. This is where you get growth factors produced. You also get this during stress or something else like that. And then, of course, this is useful in terms of mechanosensation. So, this is something that the cell utilizes as a response mechanism to different mechanical stimuli. So in the classic model, ERK sits at the core of the MAPK cascade. These Ks represent phosphorylation events. This signaling pathway that governs critical cellular processes such as apoptosis, migration, and mitosis. So the pathway is typically initiated by a kinase, RTK activation, often triggered by growth factors such as epidermal growth factor, EGF, which sets off a phosphorylation cascade, culminating in ERK activation. So you have this phosphorylation event, which is where protein is phosphorylated, and it allows it sort of like a key sometimes to let it do certain things. So, you have these multiple phosphorylation events in a cascade, and that gives us this response. It allows for this ERK molecule to translocate into the nucleus, where it can modulate gene expression programs that drive diverse cellular outcomes. So this is where, and then there are different things like during collective cell migration, where mechanical stretching activates ERK through EGF receptor signaling, generating unidirectional ERK waves that propagate to follower cells and coordinate collective motion. Tissue repair is a similar thing where ERK activation works to promote regeneration in things like skin. So this is a nice figure showing different types of ways to measure and manipulate this. So we have traction force microscopy, where we have, we have cells on a substrate, and we can get it to, move around and then image this behavior. We have single cells in a cell cluster, and we're able to image it that way in 3 dimensions and in 2 dimensions. 2D protein micropatterns, we can put this on a substrate that's a microarray, and we can look at cell migration in a very controlled way. And then, of course, microfluidic devices, which is where you put cells into a chamber, or you put molecules in a chamber filled with fluid, and you can look at the fluid dynamics. You can control the flow. You can have a laminar flow or a turbulent flow, and you can look. These are just ways to sort of, control the environment so that we can image things and, do experiments where we can remove the confounds and, collect data in isolation. And then we have to then put that together. So if we do like a single cell measurement, we have great control over that. But then we have to put that together into populations. We have to put that together into tissues and into a more general context. And this is where we have this problem of putting processes together, because we collect data this way, but of course biology behaves according to these larger structures in some sort of context. And so getting between like these measurements and that point, because we don't really we can measure things, I guess, in vivo very broadly, but the thing is, we can't control for every interaction. So our measurements are contaminated by confounds. So, sometimes it's really hard to do biology when you have a lot of confounds and then also be precise in something like biological engineering. So we use these tools in biological engineering to get a good handle on the system so we can understand how to manipulate the system. So if you've ever heard of organs on a chip, this is where they take cells from different organs, they put them in a microfluidic device, and they want to see how they behave, how they adhere to a substrate given certain flow conditions and fluidic conditions. Okay, so this just goes into applications to medicine and other types of things. This is just showing some other things about cells and tissues and context. Where you have the breast, placenta, pancreas, and breast again, and they're looking at cells from those places, and they're kind of looking at their adherence, and their adherence is going to play a role in how healthy they are. So if cells stop adhering to their substrate, they might become cancerous. It might be disruptive to the tissue, and it can lead to all sorts of problems. Okay, so then then they're really interested in tissue curvature, as we mentioned before, where you have tissue curvatures that we can model single cells and we can look at their dynamics, and if we assume like a flat sheet, maybe we're okay, but that's not the way biology is. It's very curved. So we have these these concave and convex curvatures, which are actually important in function. So in melanoma, for example, convex curvature at the tumor periphery has been shown to prime cancer stem cells, which actually results in metastasis and recurrence of cancer. So we'd like to know kind of something about the behavior of these cells so we can treat them better or treat the patient better. And so local curvature can serve as a mechanobiological cue, and it can hold the potential as a prognostic biomarker. So you can understand things about what the cell is going to do by understanding how it behaves along this curvature. You can also put cells on a curvature and get specific outcomes and predict those outcomes. In the pancreatic ductal system, curvature thresholds dictate lesion expansion direction, either outward, exophytic, or inward, endophytic. And then this is where we have these transformed cells that exhibit an apical-based redistribution of phosphorylated myosin light chain 2, which is where if we get that signal from our cells, we know that there's this cytoskeletal remodeling going on. So we know that the cells are remodeling their membranes in response to adhering to that curvature of the substrate. And so this just tells us something about what curvature is doing in biology. It's not just the geometric feature, it actually regulates the cells. So depending on the shape of the substrate, the curvature of the tissue, these are things that affect the behavior of the cells. And so sometimes curvature has a really strong functional purpose, and we need to understand how that affects the cells that are involved in that. Okay, so then they just go through some of these other things like spatial confinement, mechanical forces fueling different types of maladaptive functions, cancers, and then talking about neurodegenerative disorders. And then this part here where they talk about mechanical stress and vascular pathology. So it's a very medically focused paper. And then talking about hydrogels and how we use hydrogels oftentimes to mimic the substrate. So we can we can mediate the stiffness, we can understand if we modify the substrate, say in an engineered thing, where we populate a surface with cells, we can then understand kind of how we need to tune that stiffening to get the result we want. It's not just about replicating, the sort of the position of the cells or the gene expression of the cells. It's about replicating that surface that they sit on. So yeah, they have some other things here where they do some quick chemistry here where they're doing this sort of stuff with T cells, and they're looking at this in context in a tissue and so forth. Okay, so I think that's enough for that paper. That was a good paper, Susan. Thank you for that. And definitely, I'll put this up in the description of the YouTube video. I can put it up in the Slack channel as well. Okay, one last thing I want to talk about today. So I did do some work on some tensegrity simulations. So I was interested in, like, we've been trying to figure out how to look at different cells and their mechanics and maybe some of their tensegrities. And Susan, of course, is running into this problem of indeterminacy. So modeling things as tensegrity structures often yields these indeterminacies, these mathematical indeterminacies, where you can't model it, like say in a multiphysics platform. So one way around that, of course, is to model it sort of like a pure mechanical system, where you resolve those indeterminacies. And I think that probably it's not really the biological aspect; it's the tensegrity aspect, where you're kind of needing to balance out the different forces, and you can't do it because that's the nature of tensegrity, is it has this sort of nonlinear response. So I was interested in kind of maybe understanding a little bit about these tensegrity structures, and seeing if I can maybe generate some Python code that'd be useful. My aim wasn't to build like a comprehensive tensegrity simulation. My aim was just to take some of the papers that we've talked about in the group and then create a rag for NotebookLM. And then I was going to get it to generate some Python code. And then I was going to play with that Python code and produce some I guess they're simulations. They're just animations, really, of some of the things that are important in a tensegrity simulation, some of the underlying mathematics as well. So I trained this model on like the mathematics, all the stuff that we've been talking about, the papers for mathematics and tensegrity, some other papers. And I was, querying some things. I don't have the notebook up here, but basically I was prompting it to produce some of these simulations as outputs, or some of these visualizations as output. So this is where, let me see if I start here. This is where the first one I wrote we got some code here where it's basically defining a double pendulum. I think this is the double pendulum. Yeah, so I wanted to define a double pendulum, and I can run this. So this is a double pendulum. We talked about this as sort of an example of chaotic kinematics. So if you're familiar with the pendulum, it swings back and forth, and it's a linear system where if you put a certain amount of force in, it swings back and forth on an axis, and it produces these sort of deterministic dynamics. In a double pendulum, which is where you have one pendulum that's attached to another pendulum, you put force into the system, and it's nonlinear. This is where you get these chaotic kinematics. So watch this simulation. I'm going to run this. So,
it's kind of a three-body problem.
Yeah. Where you have the basically the 3 degree or 3 joints, and I think it's 2 degrees of, well, how many degrees of freedom is it? Is it 2 or 3? I'm not sure. Well, anyways, these are the chaotic kinematics. So instead of a deterministic swing, you get these different types of dynamics. And this is totally independent of the amount of force you put in, because you're just shoving it with a certain force. And it's just going through this, that's generating this trajectory.
Yeah, its trajectory is very dependent on its initial conditions,
Right.
which is a definition of a chaotic system.
So wherever it starts, well, it's going to depend on whatever the input is, and it's going to determine the rest of the trajectory. But other than that, it doesn't control the trajectory. It just pushes it in a certain direction, and then the dynamics unfold. So every one of these simulations is different if you look at it. I mean, they're similar, but they're different. That's the idea. Okay, so this one then is where I asked it to build this very small model of actin cables and microtubule struts. And actually, when you train it, it picks up on the keywords and the parameters, so it knows kind of what it speaks in that language. And sometimes it doesn't give you what you want at all, but you have to try different types of prompts and try to constrain it, pin it down to like, you'll ask it a general thing. You'll prompt the model with a very general thing about biotensegrity, and then you look at the thing you got, and then you'll say, okay, these are the cables and struts make these different. So you have to like drill down on the thing you get. So you have to refine it. So this is where we have the structure. It's kind of fluctuating on this two-dimensional plane, and this is just showing this sort of stability. I don't know if that's it's supposed to be super stable cell dynamics. I'm not sure it's useful, but it's like very simple. This one is where we have a helical twist. So this is a three-dimensional prism. I think that's what I prompted to create, is this 3D prism. So I prompted it to create this 3D prism, and I wanted it to twist helically. So we have our struts and our cables in red, our struts in green. And then what it's going to do is it's going to rotate, or it's going to twist at this angle. And you can see the dotted line, which is the twist, back and forth. So again, just a demonstration of it, just showing the structure.
That's the simplest 3D tensegrity you can make.
Yeah.
Yeah. Called a T3.
Yeah, T3. Okay. So then this one is where we have a 3D epithelial layer with a helical rotation. So this is where it's this interactive slider. Oops. I'll have to redo it. Okay. So this is where there's a helical twist. I can move it around. Let me see if I get it in view here. So you can see that. I mean, let me actually make this smaller so you can see this a little bit better. All right. So if I move it back and forth, it's moving, it's aligning like this. It's so at 60 degrees it's aligned. Yeah, 60 degrees it's aligned. It's coming out of alignment. At 120 degrees it's aligned. And then, so it's got this periodicity, and this is a 3D epithelial layer where the different layers are kind of rotating, and they're aligning at a certain helical rotation. Then there's this one here, which is the interactive epithelial tensegrity tower. So this one has a couple of parameters. This has a number of cell layers that we can modify from 3 to... So from 3 to maybe 6 to 9. So you can see it's gaining complexity. It's growing in terms of its height. So it's like 3 cell layers versus 9 cell layers. Actually, we can go down to 1, which is this. And then we have 2, 3, 4. So we can build up from there. Then we have their helical twist, which we incorporate from the last model. So it's moving. Now, the last model had just these epithelial cells that were rotating in free space, but this is where they're connected into this network. So we have the struts and the cables. And the black dots are the cells, so now it's rotating along this helical twist. And then we have the cell height, which is let's see how it's really modifying this. But anyways, you can play around with the slider, and it kind of rotates the structure and shows it's like flexing around an axis.
Can you change the cell height? Well, like, can the bottom one be thicker and then the top one be thinner? or a couple of them.
Let me see what that
Like
looks like.
if you had two, are they all the same height as the layers? That's what I was asking.
Yeah, I don't know. The cell height isn't working right. It's not really
Okay.
doing much to it. Yeah, I don't know. But anyways, yeah, it'd be nice to get something. Well, I mean, this is just a starting point, right? We can like modify
Yeah.
these if.
Yeah, well, I managed to make this in COMSOL, but the problem comes when you try to put them together,
Right.
attach them to each other. When you make a group and try to attach them,
Yeah.
that's the problem.
Yeah. And then finally, this one here is an interactive. They call it interactive prismatic biotensegrity. So this is where you have 4 parameters. This is where you can vary the number of struts. Oops. You can vary the number of struts here, where it's like, so you have 4 struts here, these red bars, moving up to 7, to 9, to 10, to 12. And you can see it's changing the shape of this thing. So it's almost like 2 fans kind of coming out from here for 12 struts. And then for like 4 struts, it's kind of like this box almost. But let's go to the one with the 2 fans. And then we have the radius. So the radius is shrinking down. So this is almost like, what do they call the shape? It's like an hourglass type shape. And then you can expand that out to this wider aperture here. So let's do that. And then let's go to the height. So the height is just, it's moving up and down. So it's shorter or taller. Let's make that tall. And then the twist, where it's twisting the struts, and the cables are kind of, they're introducing this pulling. So it's like pulling it close together, further apart. Well, actually, this is where there's no twist at all. So it's like it's like a cylinder. Yeah. So then the twist is introducing forces, tension and tension and compression. So yeah, that's what I have for that. I actually have one more. This is then the fixed soft biotensegrity cell, where it's nonlinear strut elasticity. This one might be a little bit harder to get at here, but you have the number of struts. Again, you can increase the complexity with the radius, where it's bigger, with the height, and it's just simulating really simple things. I was kind of hoping I could do a little bit more. I'll probably have to work with it more for that. Then you have the twist, of course, and then the linear K, which is a parameter, and then nonlinear, which is another, where it's actually changing its stiffness. So this is where it's, I think it's changing its stiffness. It's kind of changing its color. I don't know what the color represents. I'll have to go back and look. But basically we have that. So those are the simulations I was playing with. You can build on that and see what we can get.
Okay, could I try them out too, or?
Yeah, I can make
Share
that
them?
available. Just usually I can link it to you and send you the link, share it, and you can play with it.
Yeah, might be interesting.
Yeah.
Yeah, like I said, the problem came when I tried to hook the cells together to make a
Right.
tissue. Yeah. So anyway, yeah.
Yeah, I'll share the notebook maybe in the Slack, and then I can send Susan an email. So yeah, it's just actually I'll share the notebook, and I'll share there's a notebook in NotebookLM, which is the prompts and the responses, and that's messier than this notebook, which is like more refined. But I can link to both of them, the Colab notebook and the NotebookLM notebook. We have 2 notebooks. One is like where I'm executing the code, and the other is where I'm trying to figure out from the literature what that should look like.
Okay.
Okay. Any other questions or comments?
I guess I'd like the summary you did the other day of all the mathematics involved in tensegrity studies. So,
Yeah.
I'd like that too. So
Okay.
I don't know.
Yeah.
I guess I might as well continue with this project, even though it's supposed to be done.
Oh, yeah. A lot of projects are supposed to be done. You still go back to them because they're.
Oh yeah, yeah. You never drop a project.
All right. I think that's it for today. Thanks for attending, and see you next week.
Thank you.
Thank you. Bye.
Bye.
Thank you, sir.
Thank you. Bye.